Okay, so just to recap, this is what we filled in last lesson before we broke up for Christmas. And you can see the absorption of glucose, remember it's occurring in the ileum, in the small intestine. And remember, what happens is starch is hydrolyzed by the enzyme amylase into maltose. Maltose is then hydrolyzed by the enzyme maltase into alpha-glucose. And remember, the disaccharide, which is what maltase is embedded in the membrane of the epithelial cells in the ileum. Now, how the actual alpha-glucose gets inside the cell is using a mechanism called co-transport. Now, the mechanism of co-transport works exactly the same in glucose as it does in amino acids. So now, if you remember what actually happens in glucose, you can apply the same process to amino acids. So now I'm going to go back over and we'll start from our new part of the sheet, which is the second part up here. If you don't have this A3 sheet from class, I'll attach this to Teams, so you've got this as well on an A4 if you want to print it out at home. Okay, well remember, proteins are really large molecules, okay? Now, proteins get hydrolyzed. Now, the first type of enzyme you're going to come across will be the enzyme called, or the enzymes, should I say, endopeptidases. And remember, we know there are two types. There's gastric in your stomach and there's intestinal in the pancreas. And what the endopeptidases do is hydrolyze the proteins into smaller polypeptides. That creates a larger surface area for a second type of protease, which we call exopeptidases. And what exopeptidases do, they hydrolyze smaller polypeptides by hydrolyzing peptide bonds that are terminal or towards the ends of the smaller polypeptides. That releases dipeptides. Now, dipeptides are still too large to enter the epithelial cells. So now what happens is they are going to enter membrane-bound dipeptidases, which are the enzymes that will hydrolyze that final peptide bond. And we know that the monomers from these proteins are amino acids. Now, if we have a look down here, what this is showing you is this is like a visual representation of this membrane-bound dipeptidase is working. So you can see here, there's two molecules. So that here is a dipeptide. And what it's doing is it's entering the active site of this, and this molecule here is that membrane-bound dipeptidase, which hydrolyzes the peptide bond between the amino acids, releasing amino acids. And if we go over here, let's write that out now in sentences. So step one, you've got dipeptides enter the active site of, dipeptides, sorry, that should say dipeptides enter the active site of dipeptidases. Let me just Tipex that little bit out there, sorry. Dipeptidases that are embedded in the membrane of epithelial cells in the ileum. The dipeptidases hydrolyze the peptide bond even between amino acids allowing it to enter the cell. Now, the thing to remember is as well, in your gut, we assume that that's a concentration gradient. Now, it doesn't always work like that because you're constantly absorbing molecules into your bloodstream. And sometimes the concentration gradient isn't in favour of down the concentration rate, so there's always more there than inside the cell. When that happens, we have to go to a different form of transport across the membrane. We have to go to this process called co-transport, and co means with, okay? So what you've got here is the process. Now, the actual thing itself is we probably best putting it on a separate sheet next to this sheet because it's quite wordy, as you probably gathered, because the one for the glucose was quite wordy. If you've got space on the other side, use that, but if not, let's just pop it by the side here. Now what it's done, there's keys here to show you what each of the molecules are. Now let's get some colours. So I'm just going to get a sheet here just next to that so we can see what we've got. Now let's get my pens so we know what colours we've got. So this first one here, D, is a sodium amino acid. So Na plus, sodium ion, amino acid, because that's what we're transporting. Co-transporter protein. And, well, let's see which one is which. So this here is sodium, Na plus, sodium ions, and the circle here, that's an amino acid. Now, so that's obviously sodium ion there, inside the cell, and that there is the amino acid inside the cell, but I'll go through the mechanism in a minute. And then you can see down here, you've then got this A here. Now A is called a sodium-potassium pump, which is a special protein that carries out active transport. And obviously that's going to actively transport that sodium ion, because that's the bigger one, isn't it, there? And E is an amino acid carrier protein. So you've got quite a lot of different things going on there. So you've got 3 proteins, lots of processes to get your head round. So you need to be careful with this. Practice it, write it out, practice it again, write it out, get your processes, number the processes as well. This obviously, this bit hit down here, this is the capillary, the blood capillary. Okay, right, so let's start from the scratch at the top. So the first, so number 1. So this is where we're going to start down here. Number 1 is by the sodium ion down here, and that's going to get actively transported out of the epithelial cell. Okay, so number 1, so the sodium ion, or sodium ions, is actively transported out. I'm just going to zoom back on my visualizer, bear with me. There we go. Of the epithelial cell into the blood by the sodium-potassium pump. And because it's a protein, I'm going to colour it the same colour as I've done in the diagram. So light blue, sodium ion, potassium ion pump. Now what I'm going to do as well, I'm going to show you two things that you've said that are really subtle. You said the process by which it's being moved, active transport, and where it's going from, out of the cell and into the blood. So you've actually done three things there. You've said what it is, well four actually, what's being moved, by what process, from where to where, using that protein. You've done lots of stuff. You've packed that sentence with lots of key detail, which is really important, okay? So your second one, now what you've done, go back to your diagram, you've actively transported out the sodium ion out of the cell. So now what's going to happen is, you've now got more sodium ions in the ileum, which is up here, the lumen of the ileum, the actual space in the ileum, than you do inside the cell, because you've just got rid of some sodium ions. So now what you've got is a concentration gradient. So let's state that. So this decreases the concentration of sodium ions in the epithelial cell. Well, we know that molecules, ions, move down concentration gradients, okay? So then, number three, so sodium ions diffuse down a concentration gradient from the lumen into the cell. But they don't just do that on their own. What they do is they move into the cell along with the amino acid. So, by co-transport with an amino acid. So again, let's say what we've done. We've said it's diffusion by co-transport. That's the process. We've said from the lumen into the cell, but we've said with the amino acid. That's our extra bit of detail there. So we said with the amino acid, okay? So imagine now what's happened. You've now got inside your cell amino acids and the sodium ions. So now you've got more sodium ions inside the cell than inside the capillary. So the concentration of amino acids is higher in the cell than in the blood capillary. The amino acids diffuse into the capillary by facilitated diffusion with a, or through a, should I say, through a carrier protein. So that should say through, sorry, my writing's not gone very good there. So through a carrier protein. If we have a look again, what we've done, we've packed that sentence full of information. So we've said, well, now the concentration of amino acids is higher in the cell than inside the blood. It's going to diffuse down its concentration gradient into the capillary. The process is facilitated diffusion. We've packed the detail with the carrier protein. And I should have done that green because that's what colour I did it on the diagram. But it was an amino acid carrier protein. There you go. So that is co-transport of amino acids with sodium ions.